Okay, we'll get started now. We might have a few people coming in the next minute or two. Just to briefly introduce Sandra. Sandra is a postdoc in my lab, has been for about the last 2 3/4 years, I guess we could say. Originally from Spain, and she did her undergraduate and her master's degree at the Autonomous University of Barcelona, and then did her PhD, I think, both there and also the University of Sussex with Di Stevens. And then very fortuitously, in 2015, I got an email from Sandra saying that she was maybe interested in doing a postdoc, and we had a Skype call, and it turned out that her interests aligned really well with some of the stuff that I was doing, but didn't really have a person to spearhead some stuff that she's going to tell you about. And I didn't, over the course of that phone call, realize what a productive collaboration it was going to turn into, but I could see that she was enthusiastic and her letters were very positive, and so I said, well, why don't you come out here and we'll give this a try, and maybe you could learn some human genetics. And she has done just wonderful, incredible things with the data set that she's going to describe to you, far exceeding my expectations. And so I'm really looking forward to your talk, Sandra, and I don't want to take away from your time because I think you have a lot to say. Okay, so here she is.
Thank you very much for the kind introduction, to all of you for being here and for the opportunity for me to present our work today. So I am extremely enthusiastic to present 3 examples of alternative approaches that we have used in the past to study the genetics of substance use disorders, and close with the approach that I want to use next here at UCSD and in collaboration with the University of Vanderbilt. So the work that I will show you today is a result of a number of people, including our collaboration with 23andMe, members of the University of Edinburgh, Tony Clark, but above all, A. Palmer, who is leading this project here at UCSD. So it is my goal, and that of many others, to identify the genetic basis of substance use disorders. And the reduction in genotyping costs has led to an explosion of genotype and phenotype data that can be collected on large numbers of individuals. So we can now perform well-powered genome-wide association studies, or GWAS, to identify genetic variants, or SNPs, that are associated with a quantitative phenotype, for example, alcohol consumption, or with increased risk for disease, such as risk for alcohol use disorders. And GWAS has proven a very successful research strategy, and here I'm showing a classic example from schizophrenia. We can see that GWAS have been incredibly successful in scanning the whole genome and identifying specific genetic variants that may be associated with many traits and diseases. So we hope that GWAS will bring us closer to the biological mechanisms underlying disease, predicting who may be at increased risk to develop, for example, substance use disorders, and with the hope, potentially, that identified new treatments. So stepping back to my aim, which is, as I will be focusing on the first part of my presentation, that is to identify the genetic basis of alcohol use disorders. But unlike other psychiatric conditions, such as schizophrenia, as we have seen in the previous slide, or for major depression, that have identified many novel genetic loci, for substance use disorders, there has been limited success. So the field of alcohol use disorders, and led by members of the Psychiatric Genomics Consortium, PGC, the Substance Use Disorder Working Group, has made terrific success in assembling large cohorts of carefully diagnosed cases of alcohol dependence. And we now know again and again that ethanol metabolizing enzyme genes on chromosome 4 are associated with alcohol use disorders. But we're still on the hunt for genes that go beyond pharmacokinetic factors. And it is possible that the signal is already there, that it may be buried or hidden, and that we need yet larger sample sizes to make this signal pop up above the stringent significant threshold line of 5 × 10⁻⁸. But, and here's the first challenge that we're facing, and this is not unique to substance use disorders, but it is shared by other psychiatric conditions, which is the difficulty to obtain a large cohort of carefully diagnosed cases for alcohol use disorders. And the second challenge is that alcohol use disorders and substance use disorders in general are chronic conditions that change over time. For instance, there is a pronormal stage during which certain individuals may be predisposed to be exposed to drugs or alcohol, which may be followed by excessive compulsive alcohol intake leading to dependence and relapse, as we can see in this spiral of addiction. And hence multiple heritable traits may influence alcohol use disorders at different stages. So to circumvent those issues, we studied normal function in the general population. And this approach is in line with the RDoc initiative in that it views psychopathology as extremes of normal curve. So we use intermediate phenotypes. We use the scores from a questionnaire audit that measures alcohol misuse and which can be measured in the general population that is not ascertained for alcohol use disorders. And it has been shown that extremes of this curve, that is high audit scores, are predictive of alcohol use disorders. So in a laboratory setting, if we wanted to collect a sample size as large as the one by the PGC, recruiting 1,000 individuals per year, it would have taken us 25 years to complete this study, and the life of a postdoc is ever so short, you will agree. So instead, we collaborated with one of the personal genetic companies, 23andMe, that allowed us to obtain a large sample size in less than 4 months. So we administered the online questionnaire of audits, and we calculated the audit total score, which is the sum of all 10 items that we have in the screen. That is, we're using a quantitative measure. We also collected genotype data from over 20,000 research participants from 23andMe of European ancestry. And so the first question that we asked was whether this trait was under genetic influence, or in other words, what is the SNP heritability of this trait. So to address this question, we use LD score regression, or LD score, which is a method that, as many other methods, is framed under very specific assumptions. So in this case, LD score is framed or exploits the fact that under polygenicity, SNPs that are highly correlated with other SNPs, that is in high linkage disequilibrium or high LD or high LD score, are more likely to attack a causal SNP, more so than lonely SNPs, as we may call, that is SNPs that are not highly correlated with other SNPs. So on average, we would expect that SNPs in high LD blocks would show higher association test statistics than lonely SNPs. So we can regress the association test statistics of all SNPs on their LD score, and the slope of this regression line would be our estimate of SNP heritability. So as we can see in this plot, a trait under genetic influence would show a strong correlation between association test statistics and LD score, and a trait under no genetic influence would show a flat line. So we applied this method, and we identified that 12% of the phenotypic variance in audits can be explained by common genetic factors. So to put this in perspective, SNP heritability estimates here in dark gray tend to be lower than those from twin or family-based studies here in light gray. But we must bear in mind that SNP heritability estimates capture a proportion of the variance in a population, that is the population that we have studied in our particular study, and that it can be measured by the common genetic variance that we have measured in our particular study. So therefore, it is not a surprise that SNP heritability estimates tend to be lower, because we know we're not capturing all factors. But the take-home message here is that substance use disorders are heritable, and that this is comparable to other psychiatric conditions. So knowing that this trait was heritable, we next performed a GWAS of audit score in over 20,000 research participants from 23andMe. And as we can see in this rather sad-looking Manhattan plot, that although it was reassuring that among the top signal we identified variants in ethanol metabolizing enzyme genes on chromosome 4, we recognized that with 20,000 research participants, we were largely underpowered. So we did seek for additional data, and I am sure that many of you are already familiar, but UK Biobank is a wonderful research initiative that contains deep phenotypic and DNA data from over 500,000 research participants. So we identified that UK Biobank had audit data available in a sample size 16 times larger than the one that we had previously published. So we performed a GWAS of audit score in this cohort, UK Biobank, in collaboration with members of the University of Edinburgh, particularly Tony Clark. And so we next performed a GWAS meta-analysis of both cohorts, UK Biobank and 23andMe, with the aim to uncover novel biological findings associated with audit. So in a single slide, we have the summary of all this work, the Manhattan and Q-cube plots of the GWAS meta-analysis of UK Biobank and 23andMe data, which identified 13 independent SNPs located in 11 risk loci and with a nice inflation attributed due to polygenicity. And in the interest of time, I will only present a series of regional association plots with the strongest signals that we identified, but I'm happy to chat more after the talk, and more in-depth details are discussed in the recently published paper. So if we zoom in the strongest associations, we can see that the top signals fall in ethanol metabolizing enzyme genes on chromosome 4. These genes have been previously associated with both alcohol consumption and dependence, and here with audits. So this seems to be, of course, a robust candidate gene. But beyond pharmacokinetic factors, we also identified signal in psychodynamic genes. In this case, we replicated signal in the gene GCKR. This gene has been previously associated with alcohol consumption, and it has an important biological function in relationship to alcohol, as it is responsible for phosphorylation of glucose in the liver. So this again seems to be also a robust candidate gene. And we also identified novel loci, which in general we found that they mapped to highly pleiotropic genes. So in this case, we can see that many genetic variants mapped to the gene MAPT. MAPT encodes the tau protein and therefore has been previously associated with Parkinson's disease, more recently with neuroticism, and here with audits. But because of the extended LD in this region, it will be hard to disentangle where the signal is coming from, whether it is coming from MAPT or whether it is coming from neighboring genes, such as the corticotropin receptor 1 gene that has long been studied with its relationship to anxiety. So this will require follow-up analysis. So it became apparent that in order to prioritize which genes, we would need to integrate data from multiple levels of analysis, including, for example, specific tissue expression quantitative trait loci, or EQTL, with the aim to prioritize genes based on the effects of SNPs on gene expression. So ideally, we would collect brain tissue from our research participants, we would measure gene expression, and we would correlate gene expression with genotype data. And we would do so to identify novel genes associated with audit. So, of course, we did not obtain brain tissue from our research participants, but instead we used transcriptome-based analysis that allow for SNPs to be linked to gene expression profiles in smaller training samples, in this case from GTEx portal. So that way we can predict genes with higher or lower gene expression associated with audit based on our genotype data. So we use sPredict scan, which is a recent method that is wonderful for a number of reasons. First, it reduces multiple testing burden because we're only using SNPs that we know they affect gene expression. Second, we don't need transcriptome-based data because we're using pre-calculated models from smaller training samples. Third, we're using tissue-specific data, so we can use tissues that may be relevant for our traits, for example, in our case, brain. And fourth, it requires only summary statistics, and this is important because more often than not, we run into confidentiality issues that prevent human genetic data to be shared across research groups. So in fact, we use this method and we identified a gene that we may have not noticed just based on p-values alone, as I will reveal in my next slide. So here we have another strong association that we identified with audit. In this case, we identified variants in the gene KLB. KLB has been previously associated with alcohol consumption in humans, but also in mice. For instance, KLB knockout mice showed elevated ethanol consumption. So this seems to be a robust candidate gene, and we could have stopped there. But we have some reason to believe that this region may extend to upstream genes of KLB, such as RFC1. For instance, the top variant in KLB is an EQTL, that is, this variant affects gene expression of RFC1 in the brain. And S-spread scan analysis identified that lower RFC1 expression in the brain was associated with higher audit scores. So we can also prioritize genes based on gene-based analysis, and in this case, we would annotate SNPs on genes and associate gene sets to pathways. That way, we're leveraging the functional relationship of genes. And like transcriptome-based analysis, gene-based methods also have reduced testing burden because we're only using protein-coding genes rather than all sets of SNPs. And they also give a more biological interpretation to our association results. So in this slide, we can see the summary of the association results for this gene-based analysis, now each dot being a gene rather than a SNP. And as we can see, we identified multiple genes associated with audit. By to identify some, we can see that, again, KLB, CKR were associated with audit. As we expected, the corticotropin receptor 1 gene was also associated with audit. A classic dopaminergic D2 receptor, which has long been associated with rewards and drug addiction, was here associated with audit. And a very fascinating gene, CADM2, cell adhesion molecule 2, was now associated. This gene is fascinating because it is associated with cell adhesion. It is highly expressed in regions of the brain that are involved in reward and inhibitory processes, and it has been previously associated with alcohol consumption, with cannabis initiation, and personality traits that are associated with risk and sensation seeking, as I will show in the last part of my talk. So this trait audit is highly polygenic. So we wanted to exploit recent polygenic analysis to see whether risk for audit would confer risk for other traits and conditions. And so to this aim, we use LD regression score, which allows us to calculate genetic correlation to assess the biological overlap between a pair of traits. This method is also wonderful because it can be applied to summary statistics, and it uses the effects of all SNPs, even though those that do not reach genome-wide significance. And lastly, and this is important, it uses two independent GWAS studies. It can also be applied to two traits measured in the same cohort, but the beauty of this method is that it can be applied to two independent GWAS. And this is highly beneficial because it is not biased by the effects of the environment, because we are not using the same individuals, and therefore they do not share the same environment. So this method has been applied to a full battery of GWAS summary statistics, and as we can see in this matrix, substance use disorders are highly genetically intercorrelated. Just to highlight a couple of examples, alcohol consumption correlates highly with cigarettes smoked per day, and also with smoking initiation and cannabis initiation. So going back to our study, and knowing that based on epidemiological data, audit has been associated with alcohol dependence and other traits, we hypothesized that one explanation underlying such association would be due to a common genetic basis between this pair of traits. So we applied LD score, and we used our GWAS summary statistics from the GWAS meta-analysis of audit, and we calculated genetic correlations with a number of traits measured in independent cohorts, for example, tobacco, smoking, schizophrenia, and many other traits. So in this slide, as we can see, we expected that audit would correlate strongly genetically with alcohol-related phenotypes, and this is the case. We can see very highly with alcohol consumption, also with alcohol dependence, and also with other substance use-related traits such as smoking and tobacco initiation. But we also observed very paradoxical associations. In this case, audits showed positive genetic correlations with, for example, higher education. So we wanted to follow this up as we were puzzled. And we wanted to explore the attractiveness of this 10-item questionnaire, which is that it can capture both aspects of alcohol consumption and alcohol misuse. So for example, the first 3 items capture aspects of consumption, which we called audit C. For instance, how often do you drink alcohol? And the remaining items capture aspects of problematic alcohol use, which we called audit P. So for instance, not being able to stop drinking. And so we hypothesized that different audit dimensions may be tagging risk for alcohol use disorders at different stages, whereas audit C may be tagging risk at earlier stages of alcohol use vulnerability, such as alcohol consumption, and potentially explaining some of the paradoxical associations that we observed, whereas audit P may be tagging risk at later stages, such as alcohol dependence and potentially comorbid psychiatric conditions. So we calculated, we performed GWAS of audit C and P, and in the interest of time today, I will not show you those results, but show you the GWAS summary statistics that we use to perform genetic correlations to see whether these 2 traits would show different patterns of genetic association with other traits and conditions. So this slide mirrors the previous slide. All audit dimensions correlated positively with alcohol-related traits and other substance use-related traits, just smoking and cannabis initiation. But interesting findings appear when we looked at psychopathology, as it was only the audit P dimension that was positively genetically correlated with psychopathology. For example, with ADHD, schizophrenia, and major depression. And the opposite pattern of association, if anything, was found for audit C in the case, for example, of negatively genetically correlated with major depression or ADHD, suggesting that potentially there is a different genetic architecture between these 2 audit dimensions. So for the final portion of this first study that I want to discuss today, and this is a uniqueness that the approach that we have used, is that audit thresholds have been tentatively used to predict people at risk for alcohol use disorders. But here we wanted to perform a series of case control analyses with the aim to identify which threshold, if any, would yield a high genetic correlation with alcohol dependence and hence be informative for studies of alcohol use disorders. So we performed a series of GWAS case control analysis. We used 3 different controls, thresholds, and 6 different case thresholds, and we performed genetic correlations with the latest GWAS meta-analysis of alcohol dependence performed by the PGC. And here we have the results. So in this busy graph, we can see the audit case threshold in the horizontal axis. We can see the genetic correlation with alcohol dependence on the right, and the number of cases on the right. And here we can see that as we increase the case threshold, the number of cases dramatically decrease, as one would expect. And we can also see the color of the gray lines indicates the threshold that we use to identify control subjects. So here we can see, first of all, the genetic correlations were high, but that we can maximize genetic correlations when we use a control threshold of 4 and a case threshold of 12, as this achieves a high genetic correlation while still retaining a high number of cases. So this could be an alternative approach to identify people with alcohol use disorders, identify cases, and use them to perform genetic studies of alcohol use disorders to increase sample sizes in a cost-efficient manner. We're currently exploring this possibility with members of the PGC, and we will hope to test those analyses and see if we can include cases for the next GWAS meta-analysis of alcohol use disorders. So to summarize, I hope this first study, I hope that it has become clear that using traditional base cohorts, it would have taken us 140 years to obtain such high quality phenotypic data. And in fact, we have been able to discover 11 novel race loci, making a huge advance to the field of alcohol use disorders. And I hope I don't have to convince this audience that using intermediate phenotypes such as Adhd, which is a fast, easy questionnaire to administer, we can learn about alcohol use disorders, and particularly by breaking down genetic influences into specific components, such as the component of consumption and misuse. And with Tony, we have created the Adhd Consortium, and we are currently seeking samples to perform an even larger GWAS meta-analysis of Adhd by 2019, and we look forward to collaborate with anyone that may have Adhd and genotype data available. But of course, the picture is not complete, and we are currently working with 23andMe to annotate all the traits that may contribute to the spiral of alcohol use vulnerability. And one such trait is impulsivity, which has long been studied with its relationship to substance use disorders, and that it influences substance use disorders at different stages. So impulsivity is not a unitary construct. It has defined neural and neurochemical substrates. It's associated with different neuropsychiatric conditions. It has been particularly well studied in its relationship to substance use disorders. And for the second study that I will present you today, we wanted to focus on one measure, delay discounting, which is the preference for small, immediate rewards of a larger but delay rewards in time. So you can do this quick test now, but individuals showing high delay discounting would prefer $34 today over $50 in 30 days. So delay discounting follows a delay-dependent hyperbolic curve. It can be captured in a single score that describes how rapid individuals devalue rewards in a function of time. And hence, delay discounting is objective. It can be measured in the general population. It fits in the RDoC initiative because it is highly correlated with numerous psychiatric conditions, including substance use disorders. And it is conceived to be closer to psychological, neural, and genetic mechanisms, more so than the abstracted disorder categories. So we administered a delay discounting questionnaire using the 23andMe online platform, and particularly we used the monetary choice questionnaire by Kirby to quantify delay discounting rates. And we used the cohort that I described earlier. And so we performed a GWAS of delay discounting, and here we can see the Manhattan plot of these results. So at first glance, we can see the highly polygenic nature of these genetic contributions. We can see one genetic variant surpassing the significant threshold line. At a second glance, we can see that our results did not replicate previous candidate gene studies of delay discounting. If we zoom in indeed the top signal, as we can see in this regional association plot, we can see that the top genetic variant was highly correlated with other variants in the region, as one would expect on the polygenicity. We can see that this variant falls on the X chromosome on a very interesting biological gene, GPM6B, interesting because of its biology. It's associated with internalization of serotonin transport, and serotonergic signal has been strongly implicated with delay discounting. And GPM6B mRNA levels were found to be downregulated in the brains of suicide-depressed patients. So this seems to be a robust candidate gene, but we know that GWAS can only identify a region in the genome that is associated with a trait of interest. So it is standard to seek for replication, and we struggled. But after contacting many groups, we collaborated with Gonzalo Becasis, that has is leading an online academic platform, in this case, Genes for Good, and we genotyped and phenotyped a smaller cohort of European subjects. And we can see in this table that GWAS meta-analysis of Genes for Good and 23andMe data supported the original finding. Now we're still seeking replication using larger cohorts, but another way to replicate those findings would be to use animal models. And in fact, I'm very glad that Sam is in the audience. We have created GPM6B mutant mice. We have now tested them on a delay discounting paradigm, and so we hope to update you on those results very soon. So I want to also discuss very briefly the second most associated signal for delay discounting, as even though it was not genome-wide significant, we believe it is quite robust. The top genetic variant is an EQTL, that is, this variant affects gene expression of the gene EVPL in whole blood. But again, this is another case when we include multiple layers of analysis, such as with EQTL data, using Experimentation scan. We identified again that perhaps this region extends to upstream genes, such as CDK3. Experimentation scan identified that higher CDK3 expression in the hippocampus was associated with higher delay discounting scores, suggesting that potentially this could be a more robust candidate gene to follow up. So as in the case for audits, we hypothesized that the observations that we have observed between delay discounting and multiple other traits and conditions could be due to common genetic basis between these pairs of traits. So we performed genetic correlations using LD score, and the first set of questions that we asked was whether delay discounting and smoking, which have been strongly associated using preclinical and clinical models, would show a genetic correlation. So the answer, of course, is yes. We did see a positive genetic correlation between delay discounting and smoking initiation, and negative genetic correlation with smoking cessation. And even though genetic analysis cannot disentangle causality mechanisms, we view these results as delay discounting promoting smoking initiation and impeding smoking cessation. Delay discounting and ADHD have also been strongly associated at the phenotypic level, but now we show for the first time that these two are positively genetically correlated, suggesting that delay discounting could be an endophenotype for ADHD. Now, we also observed unexpected negative association with schizophrenia and positive genetic correlation with major depression. These observations have not been made strongly at the phenotypic level, so these analyses show how this can open novel areas of research, suggesting that delay discounting could also be associated with these conditions. But this, of course, will require follow-up analysis as well. So delay discounting is also associated previously at the phenotypic level with educational disadvantage and poor health outcomes. And here we show that there is indeed a negative genetic correlation between lower years of education and a lower childhood IQ, and with higher obesity rates and higher BMI. And height, which is not strongly influenced by an individual's behavior, was not associated with delay discounting, which we used as a control. So in this second study, I have presented the first and largest genetic study of delay discounting, and we have been able to identify a novel loci, GPM6B, on the X chromosome. I have shown you that the genetic signature of delay discounting overlaps with other traits and conditions, so using again this intermediate phenotype, which is very easy to apply, could help us learn about the biology of these disorders that are very hard to collect using traditionally ascertained cohorts. But we're very excited with APE, has recently received funds to expand our sample size and collaborate with 23andMe. We are hoping to get over 105,000 research participants, so we hope that by 2019 we will have discovered novel genetic loci and its relationship with delay discounting. But if you recall, I have mentioned that impulsivity is a multifaceted construct, and we've only studied one such facet, delay discounting. So in the third study, we gathered information about other impulsivity subtypes. So we administered online questionnaires to the cohort that I described before, and in particularly, we used the UPS and BIS questionnaire, which are two of the most commonly used self-validated questionnaires, and each capture different aspects of impulsive personality traits. So examples of these questionnaires could be for UPS, sensation-seeking subscale, would be I quite enjoy taking risks, and for BIS, I do things without thinking. We also gathered information about drug experimentation. So we administered a survey that gathered information about how many different drug types an individual had tried in their lifetime. So this measure could go from 0 to 11 according to the different drug classes that we used, and hence we're using a quantitative measure. So we identified one genetic signal, and it was for the subscale of sensation seeking for UPS. In this case, we identified, may sound familiar, the gene name, a genetic variant associated with the gene CADM2. We were also surprised when we looked at drug experimentation. We found a very strong nominal association between variants in the gene CADM2 and drug experimentation. The reason why we're so enthusiastic about this gene, we believe this signal is very robust, and the fact that we believe it's very robust, it is because we've also performed another GWAS meta-analysis in collaboration, and looking at a broad measure of do you like to take risk from a single item in over 1 million participants. And again, we can see that the strongest association was in the CADM2 loci. So if you recall, CADM2, I have mentioned, is highly expressed in regions of the brain that regulate rewards and inhibitory processes. It has been associated with our measure of sensation seeking, drug experimentation, audits, and in other independent cohorts measuring alcohol consumption, cannabis initiation, and risk from 2 independent studies. So this gene is potentially underlying a personality trait that may predispose individuals to engage in risky behaviors. So this seems to be a very robust candidate gene to also follow up using animal models, and in fact we have done exactly so. We have created CADM2 mutant mice. We are fortunate because using intermediate phenotypes, we can translate those human results into the laboratory. And we're fortunate because we have many tasks in mice that model risk from humans, such as the Iowa gambling task or exploratory tasks. We will also measure alcohol intake from these animals. And we're also very excited to collaborate with Thomas Vieder from the University of Tufts, because this gene is involved in cell adhesion, in plasticity, synaptic plasticity, and we want to now characterize synaptic morphology of these animals in regions of the brain, such as the frontal cortex or striatum, that have been strongly implicated in reward and inhibitory processes. For the last part of this third study, we wanted to exploit the fact that we had so many impulsivity measures and see if those correlated genetically with substance use-related traits, as we have seen this association again and again at the phenotypic level. So as we can see in these slides, we did see positive genetic correlations between impulsivity measures and, in this case, tobacco and cannabis initiation. But what is intriguing is the fact that we observed that some impulsivity subfacets may be strongly or more strongly genetically correlated with specific drugs of abuse. So for example, in this case, BIS was positively genetically correlated with cannabis, but not smoking initiation. And another measure of UPS, questionnaire premeditation, for example, was positively genetically correlated with cannabis, but not smoking. So of course, this is still in its infancy. It may attract attention to researchers in the field of addiction. This is merely speculation at this point, but it may suggest that different impulsivity subtypes may predispose individuals to be exposed to different drugs of abuse. Impulsivity is also a hallmark of ADHD. And here we can see that we showed positive genetic correlations between measures of impulsivity, particularly those from BIS and ADHD, again demonstrating that impulsivity is an endophenotype for ADHD. And lastly, with regards to our measure of drug experimentation, we did see that it showed strong positive genetic correlations with our measures of impulsivity, also with other measures of substance use disorders from independent cohorts, and our measure of audit. And we also observed a positive genetic correlation between drug experimentation and ADHD. And this is interesting because this observation has been already made. ADHD individuals may be predisposed to be exposed to drugs or take drugs excessively, and these results suggest that these two share a common genetic basis. So in a nutshell, this series of studies that I have presented today show that a role for common genetic variants for traits that have never been explored through GWAS before, such as audit as a quantitative measure, forms of impulsivity, and drug experimentation. And I have shown how these studies can help annotate different aspects of this addiction spiral. So our results show that those traits are polygenic, are moderately heritable, and influenced by many genetic variants of small effect sizes. So we would have benefits from even larger sample sizes than the ones that we have used. But as I have hinted during my presentation, we are very excited to expand our sample size substantially, hopefully by 2019. And so our results share a common core goal of the RDoc initiative that shows that intermediate phenotypes of those very easy to collect and administer questionnaires such as delayed discounting audit can help to learn about the biology of, in the case that I have discussed today, substance use disorders, but potentially other psychiatric conditions such as ADHD. And I do hope that I have convinced you that using population-based cohorts, we can learn about the biology of those traits, but in a more cost-efficient manner. But I hope I have conveyed that there has been modest success using traditionally ascertained cohorts, and that this has been partially driven by the difficulty to obtain large sample sizes. Although population-based cohorts can help us achieve a larger sample size, as I have shown you today, even so, those are expensive and somehow slow. And moreover, we're limited by a single trait or few traits. Therefore, we're missing the trajectory of a patient over time. So it is very exciting to live in this emerging field in psychiatric genetics, where now genetic studies can be performed using electronic health records and DNA data. And so EHR systems are already built. They are available to researchers, and they offer a panoramic view of the patient. Compared to traditionally based cohorts, we can obtain EHR data and GWAS data at minimal cost. They also have longitudinal data, so these platforms can enable genetic studies of unprecedented size and scope. So I now want to derive algorithms to identify people with substance use disorders from electronic health records to increase sample and phenotypic resolutions to perform genetic studies of substance use disorders. Of course, this quest is not easy. EHRs are not designed for research. Developing those algorithms is not trivial. It will take time and effort to develop and a skillful team to deploy. And I am very fortunate to have the opportunity to collaborate with leading experts in the field, Nancy Coates and Leah Davies from the University of Vanderbilt, who are pioneering the work of EHR genomics. They have access to a very powerful international biobank called BioView from the University of Vanderbilt, which has deep phenotypic data and a large number of individuals that have already been genotyped, but these are growing and growing each year. BioView is part of the IMAGE network, which is funded by the National Human Genome Research Institute with the hope to use EHR in genomics. And a large proportion of individuals in BioView meets ICD-9 or 10 diagnosis for substance use disorders, as I'm showing here, and as I will be focusing on for opioid, nicotine, and alcohol use disorders. So very briefly, I now want to identify people from the de-identified biobank from BioView, identify people with substance use disorders to use those cases to perform genetic studies of substance use disorders, and next perform polygenic analysis to see whether genetic risk for substance use disorders predicts risk for other traits and conditions such as medical conditions, because we now can do so using electronic health records. So this project is very exciting, at least very exciting for me, because it will be the first to use big data and health informatics to perform genetic studies of substance use disorders. And so we can advance or accelerate phenomics and genomics of substance use disorders in a cost-efficient manner. We will also be able to compare whether genetic overlap with substance use disorders overlaps with other substance use disorders and other psychiatric conditions, because we know that those traits are highly comorbid and associated with other psychiatric conditions, but also other medical conditions. And we also want to make those algorithms publicly available to the scientific community, because it is our aim to validate or test the portability of those algorithms to other healthcare systems, such as the well-expected, soon-to-come All of Us that Lucila Ono Machado will be leading here at UCSD. So with this, I finish. Thank you very much. It's been a pleasure for me to be able to present our work, and I would love to take any questions that you may have.
Hey, Igor's not here to tell us to use the mic. Still use the mic.
So one of the challenges with EHR data is defining the cases, but also in defining the control groups as well. So
Correct.
I'm curious what some of the strategies you have for doing that, especially considering that substance use disorders may not be something that might really come up in health record data.
Yes, yeah. It is a tough question to answer. We are part of the PsychMerge consortium that they are also trying to develop definitions for other psychiatric conditions, such as bipolar disorder. So we are taking inspiration from their studies, and I agree that a big challenge is not only the cases, but more so the controls. We can try to define sets of parameters, such as, for example, at least 5 years in the records. But another strategy that we want to explore, and this is why this needs to be a skilled team, is because we want to use natural language processing to dig into the physician notes to really look whether there's clearly no exposure or misuse of those substances. Perhaps we will be able to identify controls this way that may not be true controls. But the idea as well would be to define different definitions and see how those played out, and to test the specificity of the algorithms that we develop. Yeah, but I'm also worried.
Yeah, it's a great talk. Really, really nice presentation. Just to follow up on Matt's question, although I'm thinking more about from the cases, if there are general health records for various disorders, there may be things in there, but you know, the concern, it seems to me, would be the substantial underdiagnosis of alcohol use disorder.
Yes.
I know in our sample, we don't have medical records. We ask people, have you ever been diagnosed, but has a doctor ever told you you had, X number of conditions? And a number of years ago, the sample, we did do structured psychiatric diagnostic interviews, and about, I think it was like, what is it, Matt, 30% met criteria for a lifetime diagnosis of alcohol dependence. About 2% said a doctor told them they had alcohol abuse or dependency. Because if you think about it, your primary care physician, it's not something they get into a lot, and they often don't want to. Yeah, but I'm just saying it's a tough problem.
Yeah, It is a great point, and I didn't look at BioView, but I did look at another biobank, and we did a quick search of ICD codes, and I can't remember the exact numbers, but we got a number, and then when we looked with a quick NLP app to try to identify from notes, the number increased dramatically. So it is true that there is a stigma, it seems, in doctors to not report substance use disorders. Yeah.
The upside, I guess, is, you know, if it's in there, they probably have a very serious alcohol use disorder.
Very nice talk. I have a question about the 23andMe database. So when you show the psychiatric diagnoses, are those self-report diagnoses that you get from the 23andMe database somewhere?
Are you referring to the genetic correlations when we looked?
Yeah,
like,
for example, you showed some slides where you showed the ADHD, major depression, schizophrenia. I was wondering where those diagnoses come from.
Yes, so those generally come from independent cohorts. Normally are from, I've taken them from publicly available summary statistics, and most of them are from VGC. So these should be diagnosed properly, not from 23andMe, yes.
So the genetic correlations let you correlate with other samples, so those are not from 23andMe at all.
And with your sensation-seeking and impulsivity measures, do you control it all for age in your analyses?
Yes, yes, age is included as a covariate. Yes.
Good.
So I had outstanding talk, very poised and very informative and innovative, which is very cool.
Thank you.
I have a question about kind of about severity thresholds. Low audit scores are just social drinkers for the most part. High audit scores, well, the audit has a cutoff. Obviously, DSM-5 diagnostic criteria would be a little bit more informative and more precise about the phenomenology of alcohol dependence. But they're not readily accessible to us at this juncture. But one of the things we do know from the psychiatric and substance abuse epidemiologic literature is that polysubstance abusers have a more severe form of the disorder. And I guess my question is, it's a lumper splitter kind of question. You sort of chose to do your analyses looking at specific types of drugs, but I'm wondering whether you've also done the same analysis with polysubstances.
No, and we have not controlled for polysubstances. We don't know, and we probably should expect that these individuals are not only abusing of alcohol, but maybe other substances such as tobacco. No, it is a shame. I wish we could do those analyses, but no.
Hi, it's great talk. I got a question about the cohorts in the population. So when you use the Euclid and the 23andMe, and also when you correlate with other cohorts, do you use only the European and the Euro-American part of the cohort?
Correct.
Okay, so all those summary statistics from those populations?
Yes, and that's another limitation. We've only looked at Europeans. I've been to I think it was the WCPG, and they said, let's not do more genome-wide association analysis, please. So we've also run into this problem. We're only looking at Europeans. Yes. Okay. Well, in a way, it's good because when we do the analysis, we don't have this problem. But on the other side, we've only looked at Europeans. Yeah.
Okay. Thanks.
Additional questions about the demographic characteristics of 23andMe.
Yeah.
You can give us a sense of what that sample is looking like, and then do you expect differences when you start to know tests for associations within some of these EHR data sets?
Very much, very much. Yeah, that's a great point because the population from 23andMe, and this is also why we observe these paradoxical associations initially, because these cohorts, they don't report high drug intake. They are highly educated, highly with high socioeconomic backgrounds, so they have a very specific profile. This played in our advantage because when we perform genetic analysis with UK Biobank is exactly like that. It tends to be older individuals with high socioeconomic backgrounds, low rates of drug use. So yes, I do worry that it may not extrapolate, and precisely because maybe, and even it may help in EHR because maybe we will get more signal. In the case, for example, for Adhd, the problem when we're looking at populations like the ones that we have looked at is that we don't get much signal for Adhd because actually people are not really showing high rates of alcohol misuse. So yeah, I do think that we do need to move on to more severe populations, yeah.
Do you think all of us will do a better job of that?
Probably not.
There was a great talk. There was a question earlier, I guess, about trying to pull apart, what's alcohol use genes versus other substance use. Are there any methods available that would let you leverage, GWAS of, say, cannabis use disorder, even if you don't have that information in your sample, to be able to adjust for that and see what you're left with?
So to adjust for that, I'm not really sure, because in your population you would have to also have measures of those drugs, right? Yeah. And the problem is that to have a sample where you have information about all of this, which I'm not sure. There are methods, but I'm not sure as well how well they would dissociate signal that comes from different drugs. Like I'm thinking MTAC could be one, but even so, the interpretation of those results, I'm not so sure if it would help much. You could look at genetic correlations, but again, this is only a number, so I'm not sure if that goes deep into the genes. Another way would be to use transcriptome-based analysis, I think, and then look for the correlations at the gene level, and then see which genes are common to some and not to others. But yeah, I'm not sure, but I would love to have those methods. and even more so, I would love to have a sample that covers really deep phenotypic data on different substance use disorders, and that's why I think that EHRs can help with great complexity to clean up the phenotypes, but it may help.
I mean, to be fair, so in 23andMe, in 23andMe, we do have data about other drugs. It's just that the use of other drugs is very low in that cohort, and so maybe you come away with the impression it's probably not such a problem in that particular population. I mean,
and same problem.
I was scandalized at how squeaky clean those people are.
And same probably also will happen with UK Biobank,
yeah,
which
definitely
could be another good one. But EHR.
So I'm not sure if I read it right, but I was just curious when you were showing these different traits associated with different substance use disorders. If I read it correctly, I think perseverance was positively genetically correlated with cannabis and nicotine, smoking. And I was just curious.
Yeah, perseverance is a very strange subscale, and we believe. Let me see. Is it this one?
Yeah.
Perseverance. This case.
It's really unpleasant to smoke, so you have to persevere. Maybe.
Any sensible explanations?
No. No.
I didn't think you were being serious.
Yeah. It was also associated with higher education, if I remember correctly, which puzzled us, but this may also be a consequence of the particular population that we have studied. So I don't dare making any interpretation.
Also, some of these characteristics, there may, it may not be a straight linear relationship.
Yeah. Yes.
You know,
Yeah.
There may be kind of
Yes.
curvilinear
Correct. Yeah.
phenomenon
Yeah.
because,
Yeah.
you know, some amount of sensation seeking
Yeah.
or whatever
Yes.
is
Must be,
positive.
yeah. Yeah.
Well, actually, that brings up, do you want to say something about the sensation-seeking and age business?
Yes, actually, we tried to replicate the CADM2 signal in another cohort, and it did not replicate. This signal is one of the few that you may all agree that it seems really robust. It has come up again and again with many independent UAs associated with risk. It's a signal that I would be very confident saying this gene is implicated in risk. But the majority of these associations have been tested on cohorts that are older, such as 23andMe and UK Biobank. And so when we tried to replicate this signal in Genes for Good, it was a younger cohort. It didn't replicate. So with the analysts from 23andMe, we asked to test whether there would be any age effect. And in fact, that was the case. There was an age effect on our CADM.
Like an interaction.
An interaction. So it is very possible, yeah.
You know, one of the difficulties with one of the difficulties of interpreting the impulsivity results is that there have been several studies where they've used psychometric instrumentation to measure impulsivity and found that they don't correlate well with each other, and they don't correlate well with
behavior
in the laboratory.
Yes.
So you know, it's hard to know what this means.
Yeah,
really.
that's a great point. And another study that if we could, that we would like to do was to look at a task. Because I agree, we may see very different results. Yes.
The other thing that I found interesting is that the gene for COMT did not come up in your analysis, and there's another laboratory in the federal government that I know very well, where you know, where the COMT and impulsivity story is kind of the lab's ongoing tale. And I find that your failure to find those results to be quite interesting.
It was a robust non-replication.
That's
too.
it was the best
not even 0.05 and 20,000 plus subjects. So hard to believe it's real. My words, not Sandra's. Okay.
Thank you so much.